and you vary egg size across here from all across these ranges. All right, so if you look at that, then you find out that this is the area here. This range of egg sizes and this range of respiration rates are the larvae that will be successful. And success here is defined in terms of being able to reach metamorphosis and complete metamorphosis. So this is the range that's successful. The rest of these all die or are not successful for a variety of reasons. Right? It turns out that this is actually an example for an oyster, not a clam, but it turns out that oysters' eggs, their initial eggs, are somewhere between 40 and 70 microns. Right? So, you know, you can quasi-validate this by looking at things, you know, sort of non-quantitative types of approaches, but it does allow a little bit of checking on this. Okay. Now, this is an example of a cohort structure that was developed from that model, from the spawning events in a year, and you can see the cohorts change with length. This is some characteristics of these cohorts. So the biomass is going down as the animals age and die. All right, and you vary these various rates. You can choose whatever you want to do it, and then construct cohorts with this range of variability. Because if you look here in terms of length, now there are animals in this cohort that are going all the way from, say, 15 millimeters up to about 25, 30, somewhere in here. Right? And then you take that information, you use a broodstock recruitment relationship to construct populations from these cohorts. Right? This is all done as in an ad hoc way, post-processing. And that's how you get these age length distributions, population distributions, and population over time, or population length over time. So it's getting a step closer to reality and to where it can be applied to things. But the limitations to this type of model is that the population variability is based upon setting up this probability distribution. It's Gaussian, and it's in sort of an ad hoc way of doing it. And you can't determine cause and effect. All you can say is that respiration rate is higher, this animal does better under these conditions. All right, so the introduction of new traits or modification to existing traits is not possible in this kind of model. And what we really want to be able to do is to track genetic variability of these populations. All right, so we now look at these kinds of models, and this is what I'm calling the present, where we are now in our understanding. Is you put in the genotype back here. All right, so you do this through knowing how many chromosome pairs there are, the number of chromosomes. You can specify loci, the number of loci per chromosome or multiple alleles per gene, or however you can say it. And so now, what we're trying to, the way things are going with these kinds of models is to integrate the population into genetic processes. All right, so now you have a genetically determined phenotypically based physiology, and the phenotypic response is now modified by the genotypic constraints. And the phenotype is what's responding to the environment. So now we have an additional piece over here, which is the animal genotype, or the genetic structure of the population. All right, so the example I'm going to give here is based upon some oyster models. All right, and this goes to what I said before, is we have to understand the biology of these organisms to really do this kind of model. All right, oysters have high fecundity. Each spawning event is 10 to the 6 eggs or more. And in some areas where there's warm areas, like along the Gulf of Mexico in the United States, where it's always sort of warm, there can be multiple spawns per season. At higher latitudes, there may be only 1 or 2 spawns within a season. All right, oysters are very annoying animals in that they're protandric. All right, so they're males when they're young and at small size, and then as they get older and bigger, they become female. All right, and so you have to track, and if you're going to do a genetic model for these animals, you have to track the sex chromosome to know male, female, male, male, or female, male. And it's the female, male chromosome, or its pair, that gives the protandry. All right, so the implication of that is the sex ratio of the population changes as it ages, right? And that's a problem because the fishery targets the large animals, which are all female. So there's some areas in the U.S. estuaries where the populations are becoming all male, right? And that's not good, because you still need the female to do the reproduction, all right? And so it's an issue in terms of management. The other thing is that oysters have a high load of lethal mutations, right? And that's classic of animals that have high fecundity, all right? Lots of animals do that. Why they do it, I don't think anybody knows, but it doesn't seem to matter because some of them don't have these lethal mutations. All right, and then there's this issue that's come up now in terms of marine diseases. It's called sweepstakes reproductive success. And what that means is that they're in a large population, like a whole bunch of oysters or clams in a bay or something, there are only a few members of that population that are actually contributing the progeny that's maintaining the population. All right, so if you have an area that's actively fished, you don't want to take out all your big producers or whatever, but you don't know who they are or where they are, and who it is changes with time and everything. So this is something that's being looked at a lot now, because in a lot of these areas where these diseases have come in and wiped out the populations, the bays are repopulated, and when they're repopulated, the genetic structure has been altered because of these sweepstakes winners. They're the ones that survive. It's the equivalent in Europe in the 1000s when the Black Plague came through, and the people who survived that ended up, looking genetically, or their immune systems were different, and then their offspring ended up with stronger immune systems, if you believe what you read in the literature. Okay, so there are a bunch of things to look at here. All right, so if you put in the explicit genetics, what you can actually do is track individual alleles and end up looking at things like genetic drift and the loss of those. You calculate the effective population number, which is the number of individuals in a population of, say, 1 billion oysters. What percentage of that is actually contributing the progeny to the population? And you can look at the introduction of new genetic traits. All right, now that's important because if you've been reading about the increasing pH in estuarine systems, there are a number of papers come out recently that show that larvae of clams and oysters and other estuarine species don't do well at lower pHs. All right, there's also, and that's something of interest now, is whether there can be some type of adaptation. Disease resistance is another one, and then just general warming temperature is an issue. All right, so now that I've said all this, how important genetics is, can we actually do it? All right, well, what this shows is down here. This is from Shmeen Woe's work, who's an oyster geneticist. This is a map of the genetic structure of an eastern oyster. The red things right here are locations on these genes that he's identified as conferring resistance to a particular disease. All right, so this particular set of oysters, or the oysters from this particular area, apparently have the genetic structure that they can survive a particular disease that affects oysters along the east coast of the United States. So what you would want to know is, how would we be able to transfer this particular genetic trait into these populations? Can we modify them? All right, so these are some simulations that just to show you. I'm not going to go into detail here, but these are 100-year simulations. These are 2 alleles on the genes. Just to show differences here, this one remains pretty stable in the population, changes a little bit. This one goes up and down. These 2 become extinct after 50 years of simulation. So you can follow these individual traits, and then they go into the progeny and look at establishment of traits in populations. All right, as an example, what's shown here, this is an application that was put in. If you add the number of immigrants here and you want to establish a particular trait in the population, what this simulation is indicating is that if you change over 25% of the population each year continuously, You know, and you're putting in this many immigrants here, this tells you how many years it's going to take to establish a trait in the population. If you do it first in the larvae, just by putting in larvae, or by putting in the adults. Right? The reason the adult is higher here is because the adults spawn more than once. All right, so this is a continuous, these two are continuous. This is if you do episodic inputs. All right, the reason this is of interest is because there are large programs. All right, I showed you that there are oysters that have disease resistance. All right, one of the management strategies for disease is to grow those oysters in a hatchery and put them into the bay, and hope they become established and they eventually become the population in the bay. What this says, that's a sustained effort over tens of years to do that, to introduce a new trait into a population like this. Right. This again is just an example here of allele frequencies on individual chromosomes, and again tracking them over time. You can see some of them go extinct, some of them have large fluctuations in the state in the population. All right, so if you do look at those kinds of things, all right, how do you then do this? All right, well, this is the again an oyster, the shellfish on the bottom here, the post-settlement population, the larval phase here. The adults spawn, the larvae carry the genetic trait, they move around in the population, and then they set somewhere either on their same reef or somewhere else, and these are the ones that are moving around the genetic characteristics. So the larvae just look at as the genes floating around. Right? So what does it take to do this? All right, when you get to this level, start looking at it, you invoke a circulation model. All right, where the larvae become particles in this. All right, there's a larval model over here where the larvae grow and they have a behavior. And then there's this genetics model that sits behind everything for the larvae and also for the post-settlement population. So the larvae spawn, they move around, and then they set in the post-settlement population. And that's how the traits get introduced into the new population. So it's a fairly large structure of model that's required when you get to this level. And what's shown on these, you know, I think I probably ran out of batteries or something. Can people yet?
Yes.
I think the battery is gone on this. All right. All right. So this is an example where this was applied to Delaware Bay. The colored areas here are actual natural reefs in the bay. All right, so if you release the particles from these reefs, all right, and this was looking at the introduction of disease resistance into this bay. All right, so if you look at the particles released from these reefs, and you want to you mark the genes with the disease-resistant gene, and you want to know where it goes. What's shown here is a connectivity matrix where this is where animals were released, this is where they set. And one of the things that came out of this is if you look here and here, there's a lot of self-settlement. So animals that spawn on a reef tend to stay on the reef, but there is exchange between the reefs. But down, you know, in the upper parts of the bay and the lower parts of the bay, they are receiving inputs from other areas, but at very small rates. All right, so this says that it's going to take a long time. If you go out here every year and dump oysters with a particular genetic trait onto that reef, it's going to take a very long time for the bay population as a whole to acquire those characteristics. All right, now this is an example of looking at exchange rates in a more general way from the upper bay to the lower part of this particular bay along the salinity gradient. And if you look at this kind of idea where you use the circulation larval model to calculate the connectivities between these areas, and then start looking at how you introduce the characteristics. Right? Because I just got through saying larvae are the ones carrying the traits. All right, so now if you do that, you start saying, well, how long is it going to take to introduce the traits from the larvae? All right, well, this tells you the frequency of the introduction over time. And if you look at this, you say, oh, the larvae are not very good at moving things around. All right, so they're not really introducing these alleles very quickly. But if you look now at the adult population of